you very much for this honor and to talk in this interesting seminar. I got this question rather late, so this will be very improvised. And of course, talking about Europe is a rather macro thing. And I'm very much a bottom-up person, so I will actually talk about myself. Because I think that I will not talk about my work very much, but I know some of you, but I don't know all of you, and I need to tell you a little bit what we do. Because I also think it's very relevant for where we need to go, of course, in life science. And this is into the clinic. So what we do, very briefly, is that we have taken whole genome sequencing into the clinic. And we've worked very much. It's a collaborative project. And actually, I started out as a professor at KI, and then I was very much involved in the early days of SciLifeLab with the aim of doing genetic disease. And once this was up and running, I went into the hospital, and I'm now head of a clinic with the actual main purpose, not because I needed another job, but to do this in the hospital. So I've sort of passed all these different parts of this process. So very briefly, what we do is we do whole genome sequencing, and then we developed a concept of extracting the relevant information and putting together all the necessary expertise around this patient group that we work with, which is inborn metabolic disorders, about 1,000 different disorders that you can treat if you find them early. And then we also have the research side. We lift filters, and then we find new biology, new mechanisms. And I won't go into detail, but this is a paradigm shift for everything we do in the clinic and in the lab. And the way we have done this is really, really integrating all these different components. And we develop our own software. We have a little community sitting with the acute pediatricians, and we have programmers. And it's really, really a very creative environment. And we're building complementary teams, and we're sharing data, and it's open source. And it's very competitive to be very generous and collaborative, and that is probably what I'm most proud of, in fact. It's a very different way of working. So it's dramatic, and it's really saving lives, and it's changed how we work with our patients. Suddenly we have diagnosis, and we can do something to them. But of course, we can also understand biology, find drug targets, and it can lead very far if we take this further. But we're still very much in a bit of a proof of concept phase. And again, the key has been the integration. So I thought what could perhaps be interesting is to think, how did we do this, and why is it successful, and what is difficult? And this, I don't have these answers, to be honest, but it would be interesting to try to do this analysis. And this is, of course, extremely important. This is critical, of course. You have to have the right attitude. And I think perhaps, I'm not sure I can speak so much for Europe, but I can speak for Sweden, perhaps, a little bit. And I think we do have a material here for several reasons. I'll come back to that. This is important. This, for me personally, this has been extremely important. The concept of, well, for many reasons. One reason is that biology is technology-driven, and clinical medicine as well. We need to focus on the breakthroughs that are technological and see what we can use them for. And also the fact of actually joining forces across institutions, medical, technical, biological, as is SciLifeLab. We have competed always in Stockholm between the universities. Suddenly we found ourselves in the same room in a very, very fruitful environment in the beginning. We had both Jan and Matthias here. It was really fun to do this, and a very new way of working. That is important. That has been one of the critical things. And of course, everything in SciLifeLab is not excellent. It's not easy. It's not enough to put people together and do this, but it creates a possibility of finding the critical people who want to do new things. And the right people that I work with today, many of them I found in this environment. It wouldn't have happened without it. This has been extremely important for me personally, and I can just emphasize the importance of funding, in my case, very much the Wallenberg Foundation, because it gives me an independence, especially going into the hospital, to really, really really be able to do my own thing, even though I'm sort of finding myself in a very complicated structure. Individual sort of independence, it's been extremely important. And then I think if trying to then, this was a little bit personal, but then trying to see the patterns here, I do think, of course, we all know that we do have a culture of collaborating. And we have a non-hierarchical structure, a rather flat way of deciding things, which can be good and bad, but I think it's definitely mostly good. And we have a lot of educated people. We have an even structure, and I think it's for a nation, it's really a stability factor. And in this day and age that we are, of course, a stable society is nothing you can take for granted. And coming ahead, I think this is an advantage, definitely for us. We do have a positive view on science, and we do have people who want to take part in studies and who trust us and are positive to what we try to do. And of course, we have a well-organized healthcare. And for me, especially then going into clinical medicine, I never actually left it, but more taking really a responsibility for leading a clinic in healthcare. This has been important because we work in a very highly specialized field, which is actually nationally centralized, which is the metabolic diseases. We do the neonatal screening, for instance. So this has been the success factors. But what are we struggling with? Well, it hasn't been very easy, because you really, really have to ask yourself, what is it exactly we want to do here? And at the present time, what we are struggling with is to take this from a project led by a few critical people, myself and a few others, to something more systematic, because we will not progress unless we can do that. And it's happening, and we have very strong support from most places, I would say, now. But still, it's not entirely easy, because there are not so many people who can take on and do the same. The multidisciplinary opportunities are also paralleled by the difficulties in parallel structures. And as soon as you get internal competition, you have problems. And this is, of course, present everywhere. I think we all know that. And that's why this is so important. The culture we made, we are seeing culture crashes everywhere. But we managed to turn that into a culture of our own, which is actively working against it. But that, again, is very much of a leadership thing. So then, again, you are suddenly finding yourself not, you can't really just copy-paste it to something somewhere else, because then you have to do it all over again. So it's a little bit, how do we do this on a grander scale? How can we learn what made this possible and do more of it? And I don't have that answer, and I would love to have some help with that. And even more so, what is the model to do this? I think we all know that we need healthcare, academics, and innovation industry. But how do we organize this? And everyone agrees that it's required, but I haven't found the optimal way of doing it. I think academia, healthcare, there is precedence, and we can do it. But the rest isn't so easy. Especially, there is a little bit of a national neurosis, I think, in mixing tax-financed healthcare with the ambition of making good business. And I'm not sure how much of a neurosis it is in the population, but it certainly is in the media. And this is a problem, I think. And I don't have the model, but I think we need to find some way with an acceptance where people understand that this is a good thing for us, for our patients, but again, we're all patients, so we need this. But I'm not sure how to do that in a more systematic way. And then some smaller things, we have a lack of experts. We are too few. We have a huge need of what we are doing and delivering, and we can't keep up because we are too few people, and it takes time to educate people. And we have some complex issues. This is certainly a European thing, and we need to be more professional in supporting people like us to deal with these issues in regulation and, like, for instance, the new GDPR things that are coming. Finally, I would say that healthcare has been harder than academia. In academia, you have a lot of freedom, of course. In healthcare, we do have a well-organized healthcare on the one hand. On the other hand, we need to identify the academic parts of healthcare and treat that differently from the rest. And we need to try to not have double administration between the county council and the state, etc. We need to centralize something so we can focus on the right things. Because if you work with big data, as we already heard today, you cannot do it unless we really join forces. And we will not be able to do it at all unless we do it together because we're too few experts. So we need to, because in the big issues, I think, in healthcare, in politics, would be like accessibility, hospital beds, but you don't really hear the third thing, which is how do we harvest from the knowledge explosion and the technical revolution for our patients? We don't hear that third part, which would be solved by sort of identifying those parts and centralizing those. So we can do the other things as well, which are equally important. And also, I think we need to make people look a little bit ahead and not just at what we are doing at the short-term perspective. We have a diagnostic revolution, and we will have a therapeutic revolution, but we're nowhere near ready in the clinic for that. But we know it's coming, so we should plan for it. We should have an agenda for it. And that requires thinking 10, 15 years ahead. So those are sort of just my spontaneous points from a sort of input from where I'm standing. Thank you.
I think we have time for one question. Yes?
Jan Andersson, Head of Research, Stockholm County Council. And I really like the way you structure this, but I lack one thing, which I think is extremely important, and that's leadership. Where you see these things happen, you see leadership. And you're actually one representative of that.
Thank you.
But that is not a common domain, and that's something we should support very much, because that's when these things happen.
Thank you. Yeah, well, sure.
I will focus on Europe now, and I was also talk about GMOs, but I will start about a couple of political things. As you know, European Research Council has been a tremendous success. It was started 10 years ago, and basically it's doing the same as NSF is doing in US, but I think it's doing it better. And I know from experience, because I've been on many panels in both ERC and NSF. I still can't understand that the politicians in Brussels allowed an independent organization that was just focused on scientific quality. But when it was formed, people from Sweden was extremely important. Kalle Eldin, who is now chair of the Nobel Foundation, he was in charge for the life science section. And they said it should be scientific quality and nothing else. Actually, the majority of the European countries didn't want that because they want to have a quota. France wanted their part and this and that. But the only reason why it came to be just science was UK, because they were so strong in this. They take about 35% of all the grants from ERC. And if you calculate what they contribute with money-wise, they contribute about 15%. Think now, when UK leaves, what will happen with ERC? What happens with countries like Norway, Switzerland, and Israel is that they are a part of ERC. But when ERC has selected the candidates from, for example, Israel, the Israel state take up the bill themselves. So will UK take up the bill for 35, 37% of all UK ERC scholars? Probably not, because they have to match that against what they have to fill in for the agriculture support and this and that. And if we lose UK and ERC, it's a very clear that some of the, especially East European countries, want to have quotas, and then the whole thing is gone. That's the thing that I'm worried about. Wellcome Trust is the biggest private foundations in the world. They are about the double size of us. We are second in Europe, and Norway in Norway is about half us. But we are the only 3 science foundations in Europe. I'm a part of the European CEO network of private foundations. We never talk science. We are more focused on the gala dinners and this and that, and it's a really schizophrenic thing when you go to think. I can tell you that when the refugee crisis came, Jeremy Farrer and I, he is my similarity on the Wellcome Trust. We said that why not make a halt at this meeting? We have a refugee crisis in Europe. What can we do? What can we discuss? No, it was not up because it was too complicated. So we have formed a network of 3. And actually, we do it with our youth. And one of the reasons is that we are going to support a journal called eLife that is open publishing in life science to form this. Now we are a discussion partner with Moedas, with the Commissioner for Science. And the only reason he wants to talk with us is that he wants our money. because they don't have enough money for basic research in the European Union. I'm extremely worried for EMBL. I mean, the type of organizations we have created in Europe to share technology, education, and life sciences. If UK leaves, if they are not going to join up with EMBL and other organizations, they will lose out completely. Of course, European Medicines Agency, Anders, you're doing everything you can to get it, and I hope we can get it. But we have a guy who is much more skilled than me on the political side of this. But I think it was a bit cheaty when the people in Skåne said that we want to have it in Denmark instead of Sweden, since many people have worked for this. It's a typical Swedish thing. It will never happen in France. They would never suggest that it will be anywhere else than in France. But we have a bit of problem in coordination in this country, especially against the European Union. So that was the political side. Then I was talked about GMOs. And since most of you are not so familiar, I will take a couple of facts. 7 to 10% of everything that we grow in agriculture is now GMOs. But everything means a farm in Africa compared to a corn farm in US. If you look at intensive farming, it's much more than that. It's 4 species: corn, soy, cotton, canola, 95%. It's mainly herbicide resistance and insect resistance. There are 4 countries that account for 85%. More than 60% of everything is grown on agricultural land in US is GMOs. It's less than 10% in Asia, and it's nothing in Europe, as you know from the GMO debate. But look what happened. In 2005, Europe was behind 25% of all the patents in biotech. Asia was not existing, more or less. It was a bit in Japan, nothing in China. At that time, they didn't care about patents, I guess. Now, it's almost gone in Europe. All the breeding companies have moved their research to China or to US. There were 3 breeding companies that had research sites in around Lund 10 years ago. They are all gone. But look at Asia, and it's mainly China. And as Petter said, they are going for the food. And they're doing it in a very, very brute and blunt way. One example is this. 4 companies in the world control 75% of the seed market. And in the plant field, is like, we talk about germplasm, cell lines. You can own a seed. So for example, Monsanto owns the right to the most productive corn in the world. And then they make it semi-sterile, of course, so no one else can just produce seeds. And that's the part of the business idea, which it's a bit cheated to the people in Africa that's trying to use it. So they control the genetic material of 75% of all seeds that are grown for our food production, these 4 companies. ChemChina, which is a gigantic Chinese chemical company. Well, to start, I must also say all these 4 companies at the top, they have herbicides and then they have the seeds. But they're really going in for the seeds because the herbicides has a limited patent time, while the seeds don't. If you own a germline, you own it for the rest of your life. So ChemChina tried to buy Monsanto. Monsanto tried to buy Syngenta. US, it was actually stopped almost up on the president level in US because food production is regarded as a part of the national security. Otherwise, they would have bought Monsanto. DuPont merged, probably just in fair for the Chinese with Dow to form Dow DuPont to be a larger entity. ChemChina just bought Syngenta for close to 50 billion USD, and that is only for the seed part. So the Chinese are extremely offensive in acquisitions, patents, and research. And they're, of course, doing it because they won't have control of the genetic material. And in Europe, we are not discussing that, which I think is a big problem. So plant biotechnology in Europe has completely lost out. Mainly all research facilities have gone. But plant genetics is actually developing nicely in Europe as elsewhere. Introducing new types of cloning, somatic embryogenesis. And since we so easy can get the whole genomes, and you can sequence all the varieties, plant breeding is actually back where it should have been 100 years ago. Now they don't have to make the cross and wait. Now they can do intelligent guesses in a completely different way. Forest biotechnology is booming worldwide. And Sweden has a very strong position through a very nice collaboration between Umeå Plant Science Center and SciLife to establish a the genomes of first poplar and spruce. Why is it booming? Because there's so much money in it. If you can make eucalyptus to tolerate 2 degrees lower temperature in frost, you can start to grow eucalyptus in the whole southern US. And if you calculate the value of that, it's like 5 times if you take Ericsson every year. So if they can do that, and it will not be a discussion about because you're not eating eucalyptus. And CRISPR-Cas open up new possibilities. Jordbruksverket said that CRISPR-Cas, if you remove a gene, it's not a GMO. The problem earlier, that when we remove genes, we were leaving a bit of things behind in the genome. So Jordbruksverket said that, and it's likely that the European Union will take that. And then you can go back. Of course, it's only to remove things. But you have the possibility with fast breeding. You remove things by biotechnology, and you add in things by breeding. And of course, focus on other things than just herbicide resistance. Drought resistance, frost tolerance, food quality, biomaterials. I'm on the board of Stora Enso, who is completely focused on biomaterials to make new products from trees. But in order to do that, you need biotechnology, both in the tree, in the plant, but in the process afterwards. And the global challenge, food. One project that I really like is the Bill and Melinda Gates Foundation. They've done marvelous things for malaria in Africa and have done a lot of things here. So they have a big project to introduce the function of nitrogen fixation into corn and canola. It will never be used in US because if you do that, the calculated you will lose 20% of your production. But the point is in Africa they don't have fertilizers. So if you can do that, you make a big change. Now some of the research groups in US have dropped out because Melinda Gates got a bit bananas about abortions and start new anti-abortion clinics and so. So even if you have a really good project that will save the world, it will not work until you have a common strategy for everyone involved. So thank you.
Thank you. We have time for one question for Göran, if there is one. No? So I have one question then. So you talk about, I like the idea about the biotechnology for trees in the forest. How big of a player is Sweden in this role?
In forestry?
Or I mean in terms of at the cutting edge of doing this type of biotechnology with trees.
We are, without any competition, best.
Okay.
Without any competition whatsoever, best. But I'm of course a bit biased because it's my former research institute.
I think that's kind of why I asked you to talk about it. All right, thank you. Okay.
I'll talk a little bit basic first about the processes to make a new medicine or a new drug. And the first part, the pre-discovery part, is where normally today that's done in universities or outside the industry. If you go back many years, we had that ourselves. But to be honest, even if we put all the money, and I thought we saw 154 billion in research dollars together, it's a lot of money. But compared to all the universities and all the schools in the world, that's very little. So of course, most of these in basic research comes from universities. They'll go in, and there, Sweden is very good. I think that we have a good situation. They go into the drug discovery phase again. Normally, small and mid-sized companies today that get up, or researchers in university that jumps in and starts their own company and starts to do it. Next phase, when we go into clinical trials, I think that we are dominating totally. Then we get into very expensive things. We get into, this could be EMA instead, review, where we get the products approved, scale up, and then post-marketing. And there are two things that have to be there to be a product. First, it had to be an unmet medical need. Whether it actually solves an unmet medical need or not, that's maybe not necessary, but it must be a perceived unmet medical need. And there must be someone that's prepared to pay for this product to get this process going. And sometimes we forget this. We think that if we have a good idea somewhere, we'll actually get a product. We don't. If you look at the starting point and look at Sweden, and that's what I look at, we have a very good situation in Sweden. We're on the top of the list. We're one of the most innovative countries in the world, and we're top, at least when the European Commission looked at it last time, we're the top country. So good starting point, good base for research. We're also fairly good in the next way when it comes to clinical trials. We used to be one of the best countries in the world to do clinical research. We're still very good. Problem is that other countries have become better and better and are catching up. And this is the volume you see. It's going down. We're still very big. And I'm a little bit proud because 1 out of 4 patients in Sweden is an MSD patient when it comes to clinical trials. So we're fairly big. And this is an important area. And you can ask, why are we so big? There are 4 main reasons. We have very good quality of the healthcare system. We have personal identity numbers, so we keep track of our patients. Losing a patient is extremely expensive because the analysis the regulators do today say that if you lose 2 patients, then you have to calculate that's the worst results you can think of, and you get that in, which means that if you lose patients, you have to treat many, many more to get the same results. And you also have a long history of registries. Some areas, when you look at vaccines, for example, for cancer, like we did most of them in the Nordic countries, we did them here because you didn't have to follow up with the patients. You just had to go into the cancer registries once a year and see, have we found a new cancer or not? And the last part of it is, of course, patient trust, that actually patients in Sweden are prepared to go into clinical trials and to contribute. And I think that there's an interesting development when we look at this, and this is taken from the Karolinska Hospital, from the Sweetheart, where they actually look at data after registration and put up in the quality registers and see, can you randomize it? Because one of the issues we have with clinical trials is that they get very, very expensive when they get big. But if you could do that in normal care and just randomize there and get the same results, that would be fantastic. One of our problems, and these are very, very old data that I picked up on the airplane from the US, is that a development of 1 new product in 2006 was $1,308,000,000 US. And that's 10 years ago. And I can promise you, it's not cheaper today. And to make this even worse, it's 1 out of 10 of these products that actually gets to the market that is profitable. Another 1 of 10, you get the money back with nothing more, and the rest of them are losers. Which is a big problem if you want to invest. So change is needed. And there are 2 reasons for that. One, of course, is that it's getting more and more expensive, and the regulators starting to see and using different techniques to get the products approved sooner. The other one is internet and media. If there's a new product for a life-threatening disease, and you can read it in the newspapers and see it on the internet, and then they say, oh no, it takes another 2 years to get it approved, and then another 2 years to get it reimbursed. People get crazy and it gets into the newspapers. So there is every reason to try to get products quicker to the market. And adaptive licensing, exceptional circumstances, and 2 things that the EMA is talking about, orphan drugs legislation, lower when it's a rare disease, you don't have to have as many patients in clinical trials, and priority review. If you really have an important new product, it gets faster. But I go back to the first development part. Are we missing the gap? Because what we do in Sweden is we say, oh, clinical research, that's important. We have to get money into clinical research. Or discovery is, pre-discovery is important. We have to have basic research and we put money there. Or we have to have something else. Quality registers, we put money there without seeing the full ecosystem where everything goes together. We need universities, we need healthcare, we need industry, both small, big companies and medium-sized companies to get this going. And this is a report that looked at gaps within healthcare, but I think in school and but I think it's exactly the same. And if you take this map, there are gaps all over the place. First of all, you have the gap when you have this new discovery. How to get in to see drug discovery? How do you get that money? Because Who's going to finance that? And we talk a lot about that California has so much money or US has so much money. And we do something about that. And I know Anders is working on that. And the government have a new fund that you could get some more money. But that's a difficult one. The one is now you have an idea. You want to get on. And you want to get into the clinical phase. That's where it's really, really expensive. And then, of course, if you are uncertain, will you actually get reimbursed? Will you actually get a product onto the market? Is someone prepared to pay for it? If you don't know that, who would ever go into financing? And then, of course, when you are there and you get all this clinical data and you've done the clinical trials and you give it to the EMA or whatever regulator it is, should we actually wait for a year for them to take a decision? When people are dying and we have the data? Would you be prepared to take the risk? And then, after the product is approved, who should follow up that we actually get the effect that we wanted? If you look at the healthcare system today, we didn't really do that. We don't actually follow up with the patients. And of course, if you actually do follow up, you will get all the new ideas about maybe this is a new patient group, a new use, how to avoid adverse events, and you get to bump to new clinical phases. So something that goes around. And again, there has to be a medical need and someone has to be prepared to pay. So I have a long list of what I think is key success factors to finish this. Top-class medical research. That's an area where we are in a good position. I think that we could be even better, but we have a good start. Top-class healthcare with state-of-the-art treatments. That's where we were 15, 20 years ago. I hope we are coming back. We today look more at budget than on results sometimes. True patient involvement with maintained trust to access data. Yes, we still have the trust, but how much are patients really on board? I think there are giant steps to take there. Detailed monitoring of each and every patient, including patient-reported data. I think that's where healthcare is coming, to collect data in every single moment to actually find out, is this a patient, could we find patient group where this is really working? Could we find the patient groups before we treat them where we get adverse events? And of course, we should utilize Sweden's leadership in IT. We are very good in gaming, in real-time data handling, and we don't. The possibility to link patient data to other registries and biobanks. Here we are good today, but there are some clouds on the horizon when it comes for the commission and we think about integrity. Whereas if you ask patients, they would say, of course, as long as I get the better treatment and better healthcare. Early access to new treatments, preferably the day after the commission approval. I think that's possible. A pharmaceutical industry that is prepared to share risk, because if we're going to go there, of course we can't say to the healthcare system, you take all the risk and we take all the money. We have to be prepared to stand up and say, yes, we're prepared to share that risk. And we have to have a deeper collaboration between academia or universities, healthcare, and industry, as many others mentioned. So I think that if we could look at this list, there are things that we can do. And of course, as I work for one company and I know many of the others, I know that we in the pharmaceutical industry are prepared to stretch out our hand and share the risk as good as we can. Thank you.
Thank you. We have time for one question. Yes?
About data, I agree with you that data is the key to the future. What's your experience in the patient wanting to have their own data? And that's question number one. And question number two is, if then you share the risk and you share the benefits, and you get patient-reported data from use as a phase 4 study, if I take 6 different types of medications for my Parkinson, for instance, from the 6 different companies, Who would take a responsibility for which side effects?
I don't think that we could take responsibility for side effects as clear as that when you take many medicines. I agree. I think it is easier to think that we will take responsibility for the efficacy, that if it doesn't work, I think that deciding which one of the products that actually cause a side effect, I think that's going one step too far. Maybe we have to find a solution where we have, like we have in the pharmaceutical insurance or the drug insurance in Sweden, where we actually commonly go together and do that rather than do it individually, because then I think that would make it too difficult.
What about the data question then, on patients requesting their own data? What's your...
I think it's obvious. I think it's the patient's data. I don't think that we have anything to do that, and I don't think that we ever should know anything about the individual patient. We should only have aggregated data from the healthcare. But the patient should have all the data. I see it as the patient's data that the healthcare is allowed to handle to do their job.
Thank you.
Thank you once again.
As the Executive Director of the Koch Institute at MIT, I will mostly speak about what we or I think are some of the exciting trends in the US, or at least in our little corner of the world. And although many of our own research efforts are focused on cancer, I hope that you will see that our approach is really applicable to all diseases. So why life sciences? You're all here, so I'm sure you agree with me that some of the biggest global challenges that we face include disease as well as climate change and changing needs in energy, food, and water for a growing population. And I think you also all agree that knowledge, biological knowledge, that we gain through research is what's going to lead us all to the future. So to achieve our vision of a healthier world, we at the Koch Institute have chosen to focus our sort of attention on our mission of both early detection for prevention and early interception, all the way through finding new treatments and ideally cures for cancer. Research is funded in the US by several entities, including the government, foundations, individual donors, universities, and of course industry, which contributes the biggest share. US academic research has sort of suffered a little bit recently in the last 5 years or so, with a substantial reduction in government funding. Not just in total dollars, actual dollars, I mean, but also when you adjust for inflation, it's really gone down significantly. If you compare to 1970, when America was devoting 2% of its GDP to research, we're now down significantly. And while industry continues to see the value of the investment, of course, they mostly fund development in R&D, whereas research is really where the most transformational gains can be made. NIH funds a lot of our research in academia, and because of this reduction in funding levels now, only about 14% of applications are successful and result in funding, which is pretty low. You have to submit a lot of grants to get one funded. And although at MIT we have a higher than average success rate, still we've seen that over the past 10 years or so, federal funds have the portion that comes from federal funding has substantially decreased, and we're now probably below 50% of our funds coming from the federal government. One point I want to make here is that early stage research is even more difficult to fund. Industry as well as government and NIH are very risk averse, and yet it is our boldest ideas that have the potential to have tremendous impact. And this is where philanthropy is extremely wonderful funding to have access to, because it's a gift and therefore there are no rules and you can do whatever you want to do. It's sad that government funding has gone down so much. As you may have heard, it may get worse in the United States. combined with also some immigration policies that may be changing, we feel a little bit attacked right now. And it's especially bad when you think about studies that have looked at economic impact of federal funding, for instance, the Human Genome Project. They have calculated that the return on investment for every dollar was $141 back to the economy. That's a pretty good return on investment, not to mention the countless jobs that have been created. Similarly, in part thanks to better medicines, thanks to research, the life expectancy in the US has steadily continued to increase. And we do face considerable challenges, societal challenges in the US. It's a very big country with a very mixed population and big contrasts. So, for instance, there are big differences in quality of life, with differences in socioeconomic status, access to healthcare. A lot of American people still have no health insurance in a country where healthcare is extremely expensive. There is a big difference in access to education. There are still states where evolution is not taught, and creationism instead is what is taught to the kids in the schools. And of course, there's a lot of difference in lifestyles. And so as you can see here, interestingly, life expectancy differs significantly depending on where you live in the United States. And it doesn't just vary by state, but even by district. And while overall, for instance, in the US, smoking levels have gone down, obesity, high-fat diets, and bad lifestyle choices are on the rise. And interestingly, when you compare these three maps, where you can see basically an inverse correlation between life expectancy and smoking and obesity, and of course, those two happen to be some of the biggest risk factors for some of the most common chronic diseases that we live with, including cancer. And obesity right now, it's still number 2 for a risk factor for cancer, but it's expected to overtake smoking very, very soon in the US. So I've been thinking about cancer for about 20 years, ever since I did my postdoc. And yet these numbers still shock me. Did you know that compared to global challenges, global health challenges like TB, HIV, and malaria, cancer doesn't only cause more deaths than each one of those, it causes more deaths than all 3 combined. In the US, about 1.8 million people are expected to be diagnosed with cancer this year. And worldwide, cancer cases are expected to increase by 50% in 15-year span. And in the US, though, although more and more people are getting diagnosed, we're finally seeing a reduction in cancer cell deaths from the most common cancers. So I would say we're at a tipping point in medicine. Someone mentioned the tipping point earlier at lunch, which was funny. Our knowledge is able to inform real advances in medicine. We know a lot more than we ever did about cancer and other diseases. We are in a time of unimaginable scientific and technological progress. And I would argue that, therefore, we're really at a transformational moment. It's a very exciting moment in time. And I hope you're all feeling the same energy that I am feeling. At MIT, we are committed to finding solutions that will overcome many of our planet's biggest challenges, including climate change and disease. MIT's motto is mens et manus, which means mind and hand, basically means to apply knowledge, not just learn knowledge for knowledge's sake. And that is exactly what we do at the Koch Institute as well. MIT has had a National Cancer Institute designated Cancer Research Center, a basic one, no patients, for over 40 years. And about 10 years ago, under the leadership of our director, Tyler Jacks, and with the, also a lot of support from the MIT president at the time, MIT President Emerita Susan Hockfield, the Koch Institute was founded. And in 2011, we moved into this brand new building. We moved MIT biologists, engineers, and other cancer-oriented researchers all together under one roof to create new cancer solutions. And in true MIT style, the Koch Institute's approach is interdisciplinary. So we incorporate biology, engineering, chemistry, and so on. And about half of our faculty are biologists, and our trainees are biologists. The other half are chemical engineer, biological engineers, material science, mechanical, electrical, computer scientists, and so on, all working side by side, mixed on floors with one lunchroom, lots of joint seminars, and all on this joint mission, which is mostly cancer. And we are taking it one step further and truly merging disciplines in our efforts. So thanks to the accelerated pace of biological discovery, the expanding power of computation, as well as new areas of focus in engineering like nanotechnology, this really felt like an opportune time 10 years ago when we first sort of came up with this idea. And just like when physical sciences and engineering combine to give us computers and smartphones, we think that we need to combine life sciences with engineering and computer science, and together that will be critical for overcoming some of our biggest global health challenges. So I think most people, to sort of explain what I mean with convergence, I think most people agree that yogurt and fruit go really well together. But if you blend it, you get something that is so much more, I think, than the sum of its parts, right? And that is what we aim for at the Koch Institute. And just 6 years after moving in together, I'm simply in awe of how well it's working. When I moved in, I thought, this is never going to work. This is a huge social engineering experiment, but it's working. And so I will spend the rest of my talk giving you some examples. First, I want to mention that we don't do this alone. We collaborate extensively. Some of this has already come up earlier, and we really have drunk from that same Kool-Aid fountain. We collaborate extensively with outside partners. We believe that science is a team sport. And to accelerate progress, we need to engage with everyone who can possibly play a role in the process. So besides our academics that are converging, we partner with industry, with clinicians at the Harvard hospitals and other hospitals, as well as, of course, donors, foundations, the government for funding, and then venture firms, venture arms, venture capitalists at pharma, and angel investors to help us start companies. Plus, importantly, already mentioned here today, the general public. We need to really do a better job communicating with them, especially in America, gaining their trust and sort of making them understand how important research is. So our purpose is to educate and train new leaders by teaching them interdisciplinary deemed science, always with an eye on the end users, namely our patients. We do this through collaboration, both across MIT, with clinical centers and industry, because we don't have our own patients, with industry, and then this collaboration is intended to produce smarter medicine. We have strategically focused on 5 target areas that we view as critical for rapid progress towards controlling cancer. We still focus on very basic fundamental biology, like aneuploidy, cell cycle control, metabolism. But with the addition of the engineers, we now also have a strong translational research sort of movement, I guess. And so I will just take you through 4 of our focus areas. First is Nano Medicine. We recently established the Marble Center for Cancer Nanomedicine at the Koch Institute with KI faculty member. And I say KI, I mean Koch Institute. You have a different KI. I will try and say Koch Institute. Sangeeta Bhatia as its director, shown here top left. So as you know, nanoparticles are tiny engineered molecules that use targeting agents on their surface to home in on tumor cells, where they deliver a therapeutic cargo that was stored inside. This reduces toxicity and side effects and allows long-term release, which can be very important. Non-compliance is a big problem in many parts of the world, but even in America. And then it also allows delivery of combinations or even sequential combinations. Nanomedicine also encompasses a field of devices, which really is new, cheap, but effective methods for detection and diagnostics, and even theranostics, which is where you combine a diagnostic and a therapeutic. In terms of what are the sort of things that we can incorporate in nanoparticles, you probably know this, but we can do everything from chemical compounds like chemo or targeted therapies, all the way to nucleic acids, including mRNA, microRNA, siRNA, CRISPR components. These are also very useful for doing screens. We have a lot of efforts also going on in terms of how to make them target to different body organs in the body. It used to be all automatically going to liver. We've now got much better at getting it to the liver specifically where we want it to go. So many Koch Institute researchers are also working on novel technologies for both cheaper and early detection, as I mentioned, and more accurate diagnosis, which is really important, so we don't have unnecessary surgeries and so on. And of course, earlier detection means prevention. This is my friend Mimi. For 2 entire years, her doctors didn't believe her when she said something was wrong, and then finally, something was wrong indeed. She was diagnosed with stage 3C ovarian cancer 5 years ago at age 46. And as you probably know, survival rates for ovarian cancer are very low, and most women, for instance, that are diagnosed with stage 3, like Mimi, have a survival rate of about 35%. This number is a lot higher if it's detected earlier, but sadly only 15% of ovarian cancer is detected early. So I will give you 2 examples of labs working on early detection of ovarian cancer, but really is applicable to any cancer. So this is a system that's being developed in Sangeeta Bhattia's lab. She was working on proteases that are expressed on the outside of cancer cells. And proteases are proteins that cleave peptides very specifically, sequence specific. And so she realized she could exploit this and has created ingestible or injectable nanoparticles that are decorated with a whole ton of these little peptide sequences that are specifically recognized by proteases expressed by cancer cells. And as a result, the proteases cut these in small pieces off these nanoparticles. They engineer them so that these small particles are just big enough to get filtered by the kidneys into the urine. And so now it's pretty easy to measure their presence in the urine. So it's like a pregnancy test for cancer. It means we'll be able to detect cancer much earlier. It's easy, it's cheap, it's non-invasive. And of course, that is also critically important, not just in the US for people who have little access to healthcare, but globally. Then this here is an example from Angela Belcher's lab. She works on bacteriophage, which are viruses that infect bacteria, only bacteria. And they are very easily genetically manipulated. So she can engineer the head to bind specifically to tumor cells, whereas the tail binds carbon nanotubes. I don't know if you know what carbon nanotubes are. 6 years ago, I definitely didn't know. But there are layers of graphene that are rolled up such a way that they reflect light in the near-infrared range. So what happens if someone has cancer and were to ingest some of these bacteriophage, they would get stuck where the cancer cells are, and along that there would also be these carbon nanotubes. Then you shine infrared light and you get a signal back. Very quick. They can go now 9 centimeters deep, and they can detect tumors that are smaller than a millimeter. I believe in the clinic, the limit is still a centimeter. It's also very cheap. She's made this machine herself. She is an engineer, but still, it's pretty impressive. And she's now working on surgical ones, so that surgeons during surgery can make sure they're not missing any small tumors that might be hiding in the peritoneum. They're also hoping to now use these phage as a theranostic and adding a therapeutic with the whole setup. And by the way, Sangeeta Bhattia's lab is also exploiting bacteria. They are programming them to destroy tumor cells. So the bacteria produce cancer drugs and then self-destruct and release the drug right at the site of the tumor. So this video will show you a beautiful bacteria colony growing, and then suddenly you see this burst of GFP that comes up because the bacteria have been programmed to have a sensing molecule that realizes when they reach a critical density. At that point, there's transcription of a lysis protein that simultaneously lyses the protein, all the bacteria, and then this drug is released en masse, and the GFP allows you to visualize it. It's really pretty cool. This was the first engineered gene circuit in synthetic biology to achieve these objectives. And so, of course, we can imagine that perhaps one day we'll be eating yogurt to both detect and kill any cancer cells that dare arise in our body. Engineers and biologists are also using systems biology approaches to better understand cell signaling pathways with all the omics that are currently generating data, metabolomics, microbiomics, epigenomics, genomics, of course, proteomics, and so on. there's a wealth of information there, and there's, we need ways to put it all together and use it so that we can predict risk, we can predict response in patients in a personalized way so we can make better treatment decisions. So one of the labs, Michael Yaffe's lab, found that cancer pathways are flexible and they can be rewired to become more sensitive to treatments. They use a time-staggered combination, a time-staggered approach, and they realized that if you give 2 drugs previously approved, doxorubicin and lorlatinib for breast cancer cells, you don't get much more of an effect than if you give either one alone. However, if you give one drug 72 hours before the other one, you get a 500-fold increase in cell death because the cells have rewired and have now become more sensitive to the second drug. He's now collaborating with Paula Hammond, who's a chemical engineer, and she designs multi-layered nanoparticles specifically for staggered delivery. So the chemistry is worked out in such a way that you get melting away of the first layer, and then it takes about 72 hours for the next layer, and then maybe a little bit later there's the inner core with some kind of microRNA against a common resistance gene or something. At MIT, the hottest topic right now, this week, I think, is machine learning. With the massive influx of data in the Omics area and everything, we really need all this, we have all this data. Machine learning can help, I think. And so I just wanted to mention that this is one of the things that we are very excited about right now. Another area that I'm sure you've heard about is immuno-oncology, immunotherapy, where we stimulate our immune system to wake up and smell the cancer or go find it. Science called it a breakthrough of the year in 2013, and you may have heard that one of our past presidents, Jimmy Carter, was not doing well at all and saved. He was cured of his cancer thanks to immunotherapy. Unlike targeted therapy, which shows initial results, but ultimately most patients still succumb of their cancer relatively not that much later than with more standard therapy. Immunotherapy, although only 20% of patients respond, if they do, it can often be a lasting cure, it would seem. And so there's lots of efforts now going to figuring out why 80% of people don't respond. If we can figure that out, then maybe we can change and shift that curve to the right. And also we very much want to shift the curve upwards so that more people can benefit. Some of the work that's going on at the Koch Institute include therapeutic vaccines and preventative vaccines, combination therapy, including combinations that would sort of wake up both arms of the immune system. Some of the early experiments we've done in mouse models suggest that this is much more effective than only going after one arm. Again, we have a nanoparticle, a nanotechnology sort of theme here. This vaccine here on the bottom is a self-replicating RNA vaccine. It's like a platform. It takes 2 weeks to make a new vaccine. So they have now cured mice of Ebola, Zika, and so on in just 2 weeks' time, very cheaply on top of it. So the US Army is very interested in this technology, apparently. And then on the right, this beautiful image, you see these blue T cells with yellow nanoparticle backpacks. It's sort of like the little power capes, supercharged T cells now that go find the cancer cells and kill them because they are armed with special powers. It can either be stimulator T cell compounds or chemo-targeted therapy. So as you probably noticed, nanomedicine is sort of a recurring theme in all the examples I gave you. And indeed, it's an area of research that's very interdisciplinary and lends itself very well on a convergence model of research. In 2010, NIH launched the National Nanotechnology Initiative, and today, this year, whatever, about 100 nanomedicine drugs are already approved and 150 are currently in the clinic. So it's a very exciting time for nanotechnology. Finally, the last 2 examples I will give you are from labs that are working on devices for monitoring patients and for predicting response. So wearables and implantable devices give patients an active role in monitoring and treating sometimes their disease. So on the left is this tiny little magnetic sensor that fits in a biopsy needle, smaller than a grain of rice. I know it's comparing to US money, but if you've not been to the US, then you wouldn't know how big this is. It's very small. It can be released in the patient's tissue surrounding the tumor, and it sort of can be part of a normal biopsy, and it sort of is supposed to monitor cancer signals. It might measure O2, pH, and things like that. On the right is a much more sophisticated device. It has about 50 small reservoirs, and each one can be filled with a different drug or drug combination. And so when somebody has cancer and you're not sure how to treat them because it's not an obvious answer just based on the genetics of their cancer, for instance, you might insert a few of these because tumors are heterogeneous, and you leave them in for 2 days, and then 2 days later you take them out with a slightly bigger needle, so you take surrounding tissue out along with it, and you stain it for markers of apoptosis, and you look which well gave rise to the most cancer cell death. It's been remarkably successful. It's in a clinical trial now, and it's very exciting. I think there's lots of efforts also going on into edible electronics as well as technologies for liquid-based biopsies and single-cell analysis. And I know I have 2 minutes left. Just want to give this last examples. Growing cells accumulate mass. If they respond to drugs, start dying, they lose mass. So one of our engineers created this little diving board that measures, just like if you have a big person on the end of a diving board at the pool or a baby, it will vibrate differently. Well, you can use that to measure the weight of cells at the attogram scale. Is 10 to the minus 20 of a kilogram. He has multiplexed this, and now he has a serial SMR, so he can follow one cell over time, over about 2 hours period. Again, you see the device is very small. Each color is a different moment in time for the same cell. It's now in CLIA approved lab space in collaboration with the Harvard hospitals across the river to predict which drug patient responds to different drugs, so you can decide which one is most likely to work. At MIT, we feel a societal obligation to move our discoveries and ideas into the clinic, and we have a number of ways to do this. As I mentioned, we collaborate with researchers and clinicians through at Harvard and beyond, but at Harvard is with a partnership called the Bridge Project, philanthropically funded. Only 5 years old, but already 5 teams of the first 20 are close or in the clinic, and we also interact a lot with industry. We are very nicely located in Kendall Square. The Broad is across the street. There's 120 companies in a 1-mile radius, and then, of course, the Harvard hospitals, Harvard University, all very nearby. Our 50 faculty have started over 50 companies since we were founded in 2008. 7, actually, this only shows from 2008. So in just 10 years, it's pretty impressive. And over 10 of those are now in clinical trials. So just in conclusion, I would say that with patients as partners and our growing biological knowledge and our expanded ability to collect data and use it to make predictions and treatment decisions, I believe healthcare solutions will more and more include participatory, predictive, personalized, and preventative medicine. All of these benefit from diversity. I mean, diversity of people, diversity of skills and approaches and technologies will bring new discoveries and tools to fight our biggest challenges. So, thank you for listening.
Thank you for a very impressive display of the power of KI. So there's time for questions. Yes, of course.
I come from KI as well, then.
Yeah, I know.
And it's very opposite, and of course, because we are a single faculty university with its advantages, hopefully some advantages at least, and maybe some disadvantages. So my question to you is then, because you present, of course, in this interdisciplinary environment, you can create a lot of added value. But will it be difficult to, in the long term, maybe maintain excellence in all areas because you don't have critical mass in all areas? I mean, so in the short term, you win, but in the longer term, maybe not so. You must have discussed this.
Yes, I mean, so we still have our original biology faculty, including some of our Nobel laureates, and you know, I would like to think that we will continue to do well in biology, but it's true. Sometimes I wonder, but maybe it doesn't matter if you're not an expert in everything. If we can be an expert in convergence, maybe that's enough. I don't know. It's a great question. It's, I mean, our students and our postdocs still come trained from places where it's very different, and it is more siloed in terms of what their skills and sort of approaches are. And so we will always have this influx of biology, pure biology students, pure BE, biological engineering, chemie, and so on. So I don't know, perhaps it will be okay.
So I'm Daniela Rhodes from NTU. So immensely impressive, but how were you able to fund this? I mean, where does the funding come from to enable this?
Yeah, sorry, so I probably went over it too quickly, but a lot of our funding is from the federal government, NIH, NSF, NCI, DOD, DOE, even sometimes some of these engineers are also working on new fuel models and converting carbon dioxide into methane and, you know, amazing, crazy things, right? Batteries made out of bacteriophage. And so a lot of our funding comes from the federal government still, and some of it comes from industry by partnering with industry. They sponsor research. In return, they get first option to an exclusive commercial license as well as non-exclusive research license. And then we have money from HHMI. A lot of our faculty or HHMI investigates Howard Hughes Medical Institute. We get money from, there's a lot of private foundations in America. So Bill Gates gives a lot of money to us. He's very interested in the third world and non-compliance issues and birth control issues and, you know, platform technologies that will be cheaper and more effective when there's not a lot of money around. So we have a lot of money from the Gates Foundation and a lot of other foundations, as well as philanthropy from individual donors, like the Bridge project I mentioned. Somebody just, you know, various donors have given us millions in these past 5 years to bring this to fruition. And, you know, it's sort of like a juggling act a lot of the time. You know, we get a grant, great, we can rest a little bit. Oh, we didn't get it, let's go find money elsewhere. We're forever fundraising.
No, I suppose what I meant was more, you know, do you get funding centralized to the Koch Institute, which you then allocated, but it's from grants. What you have said is people apply for a project to do a collaboration or collaborative project and then get funded on that rather than directly to the institute.
Yes, I'm not sure I understand your question. So a lot of our faculty will write a grant to NIH, either together or collaboratively.
But does Koch have its own pool of money that
the Koch has a little bit of its own pool of money. We fundraise a few million a year. Maybe a little bit more, it depends on the year. And we have a very sort of fair way of distributing that money, where we invite proposals once a year from all our faculty. And then we have a review committee of about 8 people who have not applied. And they each score all the grants. So every grant is scored by 3 people, and then the top scored grants get funded. And we give a lot of priority to interdisciplinary collaborative projects or junior faculty to, you know, because for them, a $100,000 grant for a year is much more important than for someone who has an established research program with a lot of funding.
I have a question about that Donald Trump has said he's going to do some cuts, and what I understand among all, NIH will be cut by 20% if it's passed through Congress. What will happen?
It's a very good question. It's one that keeps most of us awake at night. But then, you know, it was going to be cut by 18%. Some people said, oh, that he's just a businessman. It's a bargaining thing. All along, he's planning to cut it, you know, 10%. So he's saying 20, and then when you get 10, you'll be like, woohoo, we won. But actually, that was his plan all along. I don't know. Even a 10% cut would be really, really bad. But then they just passed a new budget, and there's a $2 billion increase for this coming year. And he says he's happy about it, so I really don't know. I just hope that in 2018, when we have local elections, we will change the House and Congress. And
confirmed biological mystery.
Yes.
Okay, are there any more questions for Anna? If not, I have one short question then, and that is I couldn't help but think about Ulf's presentation where he showed the plot of so many lead compounds that start off as potential leads, and then you have the funnel, right, where it winds up being only one in the end. It was something like that. I'm paraphrasing a bit. Now, when you show these really impressive early stage results for nano biotechnology, Is there a similar funnel, do you think, for that type? Or is the funnel wider? I mean, is it more likely to succeed? Or do you have any insight into that?
I'm definitely not an expert to answer that question. I would say that some of nanomedicine has been giving old drugs new life. So some drugs would be great, but their toxicity is too big and they have to be discontinued. If you can package them into a nice little nanoparticle and then you get local delivery only where and when you need it, and you don't get any of the systemic toxicity, then in a way, you haven't found a new drug. You've just used an old drug and made it new. And it probably get approved much quicker. Similarly, now that liposomes have been approved already, as long as you use the same thing, the approval should be pretty quick. Maybe cost will be less. And I think also with genomics and personalized medicine, people are getting very savvy about having smaller clinical trials that cost less money and have better, I spoke Flemish for a moment there, better patient selection. So your clinical trial may actually be more likely to be successful because you know exactly what patients you need. You're using those patients. You're likely to see a result in those patients. Before, if you have too many patients, the good results get diluted out by the bad, and then there's no benefit and it doesn't get approved and so on. So I'm hopeful that we will see a speeding up of approvals because...
a benefit at every step of that stage then.
And so I can't really answer your question. I think a lot of the nano things are maybe slightly more easy to get approved. It's not like they're completely new drugs often.
Right.
It's just a new way of packaging it.
Okay, so I think we should thank Anna again. Right, so the idea here is that we want to talk a little bit about the trend. You know, we had 3 presentations discussing trends in Europe and Asia and America. And we have a panel of PhD students that are here that are very excited to be here and learning quite a bit, as I am as well. And we've talked about some kind of starter questions that we can get the ball rolling. And the idea is that you guys kind of give your perspective on these questions. So whatever comes to mind, right? So which one of you, so maybe the students, I can say we have some from KTH. KTH contingent is here. KI contingent is here. SU contingent is here. And Uppsala, we don't really, I mean, they weren't invited, actually, sadly. Okay, so we have from a few universities here. So one of you can start then.
Yes.
And you can introduce yourself, please, yeah?
Definitely. Hi, Emma von Wedding, PhD student in protein technology from KTH. So thank you all for your interesting talks, and also thanks to the organizers for inviting us to be a part of this interesting meeting. So we touched upon the subject a bit earlier about education, but I'm wondering, like, what are the trends in these different areas regarding education versus research and the funds and efforts being directed towards them? And are there any global differences between the balance of these 2 areas?
Maybe I'd like to hear from Nan Yang, actually, because this is something I know very little about, how the funding situation is there for your university.
So my impression from Singapore is, of course, the Singaporean government has invested very, very heavily in education. You know, the students that come out of Singapore do very well in all sorts of type of competitions. As Bertil mentioned, I'm not so sure, coming from Europe, if I really, it's very few of them are knowledgeable but not very imaginative, so I'm not sure they got it right. But then nevertheless, students are, you know, they are very, certainly NTU is forever discussing how to better improve the education, and we are going towards team learning, all the, you know, all the lectures of video, the students get the lectures before they attend the lectures, and faculty works extremely hard to educate the students. In terms of research, we just had in research funding, the last year we announced next 5-year plan for research, and the funding, Bertil didn't mention this, but the previous 5 years, the funding in Singapore was 16 billion, and they raised it to 19 billion, which I think is totally amazing, you know, from the European and American perspective. So it's a new country. it's still completely determined to be best, and they're willing to invest. NTU also, Bertil didn't say that, but NTU is the fastest growing university in the world, both in number of students and also in terms of research. And they are forever creating new departments. As we said, the School of Biological Science is 15 years old, the medical school is 4 or 5 years old, and I know they are now starting a pharmacology department and so on. So it's still, I mean, it's amazing, really.
Yeah. Maybe we can ask Anna down there at the end here. Oh, you have your microphone. Yeah. Okay, because I think that I can twist the question a little bit, because I was thinking when you talk about the Koch Institute, for example, and this idea of multidisciplinary research, that's a prevailing theme today. I think everyone is very proud of their multidisciplinary. How do you train a student to be a leader in a multidisciplinary team? I mean, you said you have people coming from different backgrounds. How do they get trained in your eyes?
So we have both undergraduates and graduate students at MIT. The undergraduates mostly have lectures in their individual departments, and so they are still trained in a sort of more siloed way. One of the differences in America, which I think is brilliant, is that you don't have to decide what you're going to study until you're 2 or 3 years into your undergraduate degree. In England, you have to already choose your A levels so that you know which degree you can apply for, and then you get into that, and then that's it. That's what you're doing, like it or not. I don't know if at 16 you really know what you're going to find exciting and novel and is going to be the right fit for you. It might depend just on who your favorite teacher is. That's not a real, measure of what is good for your personality. And so the undergraduates are still trained by sort of the specific courses that they choose to do, but they spend a lot of time in the labs at MIT. There's a huge emphasis on undergraduate research opportunity programs, and so some undergraduates will have nature papers as an undergraduate because they spend 12 hours a week every week of their whole 4 years in a lab and really had their own projects and learned a lot. And they can come and partake in all of our seminars and so on. And then for graduate students, you still get hired by a specific by a specific department. So you're a graduate student of biological engineering or of biology and so on. But then when you pick your lab, you can sort of pick a lab in any department if you decide you'd rather be somewhere else. Or you can pick a lab at the Koch Institute, which is not a department in and of itself. And within there, before you pick your lab, you do rotations, they call them. You go to different labs. You can often be mentored jointly by 2 different PIs, so you could have a biologist and an engineer. And even then within the labs, a lot of the labs will have someone from biological engineering, someone from chemical engineering, a biologist, a computer science, a few clinicians. And so within the lab, everything becomes really mixed together. And then we have an annual retreat, we call it, where we all go to Cape Cod and every lab presents. And I think about this convergence model that we have, because I try and sort of talk about it to so many different groups. And we had people present from all the different labs, and it wasn't until the very end when someone pointed out that we had done biology engineering lab, that it occurred to me, because everyone now speaks one language. I don't even know anymore if somebody was originally trained as a biologist or an engineer. They just, they talk about autochthonous mouse models to study the latest technologies or discoveries in, and I don't know. And it's sort of really amazing how they've become multilingual or even, and have developed this new language. And so I think that's how we train team science, because everyone is getting really exposed to everything. And our faculty start collaborating. They hear each other's work, they say, oh, we should collaborate, and then they realize they're already collaborating. They didn't know it because the students started collaborating, and that's just wonderful when it's organically from the bottom up, ground up, whatever the expression is. It's really marvelous to see, and I think it's a bit of the entrepreneurial culture at MIT and in the US, and sort of there's no failure, no fear of failure. It doesn't matter, the only way to fail is by not trying something. But if you try and it doesn't work, well, at least you tried. That mentality is very different. I'm from Belgium originally. It's so different in Belgium. I think you are too. No, you sound a little Belgian anyway. So it's a combination of. Sorry.
No, it's okay. It's your perspective then, Anna, from Karolinska.
Well, talking about how we work systematically, there are people in the audience who know a lot more than that. We have our president or vice chancellor, for instance. But I can tell you that it's important, and what the bottlenecks we have is lack of expertise. So it's a vital question. So that's if I can turn it into that question instead. And of course, it's a problem working in the clinic that it takes a very long time to become a clinical specialist. Right. So we need to form new disciplines, and there are new areas emerging. We have clinical bioinformatics and a lot of new molecular subspecialties that we need to identify. So we need to rethink, because we don't have time to wait for the young people to train all this way. We need them now. And on the other hand, we have many young people who are just natural talents at working like this. So I'm very optimistic, but we need to take care of them, not put them in these extremely long tracks before they're done.
It seems that that most of these really successful laboratories, they do have a bioinformatician, they have a resident biologist, they have a resident engineer. You know, these are the disciplines that have to come together in a lot of these laboratories then.
Yeah, but many young people are natural programmers or bioinformaticians, and to them that's just very natural, and then they're ready to address another problem. So I think we need to see that they are sort of inherently capable of things. And
is that something that's happening in Sweden, that we're training people to be at a young age programmers, for example, so that they can contribute at that level?
Yes, absolutely. And we have many people coming out from, studying computer science who want to do biology and who want to do clinically relevant things. So, and they don't want only to go to Google and Spotify. They want to do other things, at least for a while.
Yeah.
So I think there is, it's sort of happening.
I never thought that you would be competing for talent with Spotify and Google. I mean, it's kind of
well, they can choose, and then sometimes they think it's very cool to sit with us in the rounds and see that they are saving lives by something they have programmed. And that's also a motivation for people, because many people want to do important things. Not that Spotify isn't important, but
but it's visible. What was Göran saying? It's very visible, Spotify, right? Would you like to have a final word, Daniela?
I mean, what I've seen, so when I was in Cambridge, I worked in an institute, and now in Singapore, we created an institute. And it seems to me institutes actually provide the right environment for this multidisciplinarity. Because some university departments tend to be, if you're biochemistry, you teach biochemistry. If you're engineering, you teach biochemistry. And that's really the institute provide the framework for interdisciplinarity. And the student who come in, they can walk between, they can do experiments in the lab, but they can also then go to the neighbor and do computational studies and so on. So maybe this is the structure under which we work, right? That's very, very important.
Yes.
So this sounds like it's happening in Europe, Asia, and the US. This seems to be a quite common approach then.
Yeah.
What else you guys got?
Hi, I'm also from KTH. My name is Rezan Giler, and I'm a PhD student in protein technology. So my question is regarding a hot topic in many scientific fields today, and it's the rising issue of science denial. So if we talk about alternative facts, we have seen a lot of great speaks about vaccines and other things, and they don't really work if nobody uses them or they refuse to use them. So my question is, as scientists, where does our responsibility end? Is it just generating this knowledge, or is there something else we need to do? Generating new alliances, or
Maybe Anna can answer first, since you maybe see this on the news quite often. We should not pick on the U.S., I guess, but I'm sure we all can comment. That was needed.
It is definitely challenging. I think I mentioned it. I think it's really important to connect with the general public and lay audiences and really make sure they learn to trust science and understand what's at stake and understand what we do. And then, GMOs have a lot of bad reputation, deserved or not deserved. I happen to think GMOs are just fine. Maybe some companies are not. I think there's also a role for scientists to take more of a lead in politics, I think, and try and sort of help teach policymakers about how important vaccinations are. I don't know. It's hard to say that it should be obligatory, but in Massachusetts, where I live, I think the schools have they have vaccination requirements, and unless you have medical reason not to do it, because you are a cancer patient, for instance, and are undergoing chemotherapy treatment, but you just don't go to school. Then you have to be homeschooled. You cannot go to a state school or I think a private school, and so I think there's different levels. But I personally think scientists have a responsibility, have a role to play. We actually at MIT try very hard to connect. At the Koch Institute, we have public evening lecture series events, we have children events, we have school groups that come every week and visit. We have a public gallery with beautiful images. I don't know if you saw my first slide, to really try and entice people in and make them think about what we do. And sort of MIT has this open campus, not a single door is locked. Anyone can walk in. So it's very much so that nobody thinks there's secret things going on behind the walls that are, science fiction-like. And so these big images that we have is a competition, actually, in collaboration with Welcome Images in London. And we exchange an image every year, and we're judged for them. They come to us and so on. But the idea is that it sort of is windows into our worlds of research, so that, again, it's an opportunity to tell the stories of how amazing research is and what it has to offer, and what it brings society and humanity. And I think if we can convince people that what we do is sort of earnest and full of integrity, then perhaps they will realize vaccinations are important.
And Wedell, you mentioned privacy. In Sweden, we have quite a high trust for medicine and this type of stuff. Is this a problem in Sweden with people refusing to give their data? Or?
Well, there are different parts of this question. I think we're relatively spared when it comes to alternative facts, and people are sort of well-educated and believe in science. But there are other issues coming, which is, I think I touched on when it comes to regulation, and data sharing will be essential. And Göran mentioned GMO. We don't want to end up regulating away all the fantastic possibilities we have with new science, new gene editing technologies, for instance. In that way, I think we are the only ones who can really show the benefit, why this is important, because you can be a professional regulator and find all these problems and then make legislation which just hits us. Yeah. So we have to show people why this is actually very important. And I don't think anyone else can do that. So that's...
Does it feel like there's more regulatory hurdles in Europe than, for example, Asia? Danielle, maybe you can have some experience here.
Well, I only have superficial sort of what I see within the university, and I know some, but not many Singaporeans. We Europeans somehow we dominate the faculty, so we talk to each other rather than the Singaporeans. But I see respect for doctors. I mean, one of the big businesses actually in Singapore is medical care. They have lots of private hospitals and lots of Asians fly in to get private medical care. But they don't mind. They go to the Chinese doctor, to the Chinese. And in fact, the most successful course in the School of Biological Sciences at NTU is traditional Chinese medicine. Okay. And at the same time, across the road is the medical school, the new medical school. So I think, and in terms of educating the public, I'm very surprised the university does not really reach out to the public yet. Maybe they will have to in future. Yeah. But in England, I have seen it. I mean, the same problem in the UK with contact with the population. So Cambridge has taken a fantastic lead. They have this incredible popular Science Week, where thousands of people come, and they're actually aimed at the children. Mm-hmm. Every department invites people in, and it's over a week, and they have hundreds of things. In the institute I was in, the LMB, we have open days. We have actually an office that people go out and talk to the schools and so on.
But not
to educate the public. No, in Singapore we don't have that.
Riccardo Diamanti from Stockholm University. There is, especially here in Stockholm, we've heard a lot, but this is happening a bit everywhere in the world. When it comes to research, how can university make sure that there is transparency, especially in the results? We know in Stockholm there's been some big scandals lately, but this is happening a bit everywhere. So what can we do a bit more? What can universities mainly do to maybe keep an eye on each other's, or the state maybe? What can happen from a larger point of view?
So of course, if you collaborate extensively, it's a lot harder to hide anything because it's going to get found. So I don't know. I think journals have a big role too. I think some journals now are actually asking other people to repeat experiments, and it sort of depends a little bit on the promotion system and the funding system, right? If things get really, really competitive, where you are worried about your lab survival, maybe it's more tempting to make things up. I don't know. It's a problem because it gives science such a bad name, and then at the same time, there's so much thought about how you have these hypotheses in your head, and it's so hard not to make the data fit, in a completely honest way. You see a result, you think, oh, that fits with what I think. And it just takes a different perspective to say, well, but it fits with this as well. But you might never have thought of it. And so I think it's another reason why collaboration and interdisciplinary science is a great way to go, because you will get those different perspectives.
Is there the trend to publish open access in Asia?
I mean, there are a bewildering number of journals coming up, and I think the university is starting to try and educate us, because I think we as, I think I get 10 invitations a week from new journals where you have no idea of their integrity in the university.
Right. Maybe open access isn't the best word I was looking for. Maybe I meant this transparency where all the data is also uploaded, for example. I mean, some funding agencies in Europe, they require this, or also in the States, I think that it's required to do this. Is there a similar requirement there?
Yes, so because we have also had similar problem to the Karolinska at our university, they have taken this very seriously, and I think what they want to implement is that all data needs to be stored for 10 years. What they have not figured out is how you do this. But I think that's...
Well, it should be easier with the digital era.
Yeah. No, I mean, all the students will have digital notebooks and, but it's still bewildering because it's the amount of data we create nowadays, how do you store it for that length of time? But I think people are aware. And I think there's also awareness that we really need to educate. It's a culture you have in particularly when you do research in the lab, and also educate young people, when it is, becomes fraud. Like we all know how to fix your figures to look prettier in Photoshop, but at what point does that become fraud? And I think that we need to take a step and educate young people. I mean, I used to say in my days we couldn't do this because you just took a photograph or something, that was it. You had no way of manipulating it, right? And so something we need to really, and we are becoming more and more aware of it.
Anna, do you have any comments?
Well, I agree with all of these things, of course. You need to have high standards, good systems, and transparency and collaboration, and you can never prevent it entirely because science is about individuals, but that will never pay off in the long run anyway. So,
but it's often, I mean, I think of you in particular when you have this clinical research. I mean, there it feels, somehow it feels even more egregious for this to be, because it feels so close to a person's life in a way.
Yes, of course.
I work with the different fields, so it doesn't feel that close to people's lives like that.
Yeah, but fraud is
I mean, my life, but my students, I guess.
No, I think the answers to these questions are obvious, and I think you all mentioned it, so I won't repeat it.
Yeah, right. Okay.
Maybe one could add that from a Karolinska perspective, we have to store data for a long time. It has been there all the time. We have a computer system that is valid. We have the whole system works, but the people that failed I mean, they were not using it, and the ones who should check that they use it didn't check. So, so we have the format. I mean, Karolinska has everything. It just that has to be. We have to follow it.
And learn by
it's for the department heads to really look into it, and the scientists and the colleagues of the scientists, as you said, collaborators. I mean, they were collaborators with some of the ugly cases we have had. So.
Just to make it clear, it wasn't an attack. Absolutely, it was just a normal question because in that case it came up that people's life were, of course, and then were damaged, and then it came out. But there are probably
many more cases in other fields that are not discovered. So that was mainly the question that I wanted to address.
No, but I just wanted to say that it's important to understand that we have a functioning system. It's the persons that fail.
That's often the case.
Okay, on that somewhat somber note, I guess, we will conclude the panel discussion. I think we should thank our members here for coming up. Thank you.